Great. Great. Hello, and welcome to our second public lecture of 2020. So this year, the Geological Society is celebrating the Year of Life, and this is our first official Year of Life public lecture. We're now fully in the swing of Year of Life, and as you may have noticed when you were getting your tea and coffee, today is the opening of our exhibition entitled Graphia and Friends by the artist Melanie Ewer. The exhibition is inspired by Melanie's own Jurassic fossil finds in Hertfordshire and conveys geologic time and ancient life forms through quilted work, mixed media canvases, and hanging installations. Just so you know, some of the pieces from the exhibition are available to purchase, and you can do so by filling in one of the purchase forms, which are found in the lower library. Melody came to our first lecture and enjoyed it very much, and thinks you'll enjoy this as well. We're also pleased to note that our lecture today is co-sponsored by the Palaeontological Association. PALAS, as we call it, was founded in 1957 to promote the study of paleontology and related subjects in the UK and beyond through academic journals, field guides, and meetings and events. This is the first year of their Innovations in Paleontology lecture series, with a series of talks given by our inaugural PALAS exceptional lecturer, Dr. Stephan Lautenschlager. The series from PALAS aims to promote paleontology to the wider community and to recognize excellence in research among paleontologists. Stephan was selected in a competitive process to represent the association. So tonight, Stephan will share with us how recent technological advances have revolutionized the field of paleontology. Advanced techniques such as CT scanning allow scientists to study fossils in unprecedented detail, and modern computer simulations and biomechanical analysis techniques are offering insights into the behavior and activities of these extinct creatures. Stephan is a lecturer at the University of Birmingham, and he holds degrees in paleontology, geology, and software engineering. So his work focuses on using digital simulations and virtual reality to explore the life of ancient vertebrates, including dinosaurs, early mammals, and saber-toothed cats. Please join me in welcoming Stephan today.
Thank you very much, Alicia, for the introduction and also for the invitation. Great pleasure speaking here tonight. Good evening to all of you. I'm very excited that you will be joining me to take a look inside the heads of dinosaurs. Dinosaurs such as this one, and I'm pretty sure every one of you recognized that this is a dinosaur, which is very remarkable because none of us has ever seen a dinosaur or a living dinosaur before. But still, we immediately recognize that this is a dinosaur. In this case, it's Allosaurus, a theropod, so a meat-eating dinosaur from the upper Jurassic of North America. And also, we instantly recognize that this is a dinosaur. What we usually see from dinosaurs is this, so just the hard tissues, the hard parts. Mineralized bone and teeth in the case of vertebrates, and in the case of invertebrates, mineralized shells. That's what we have as paleontologists to work with. All the soft tissues, which might be very interesting, but have not preserved. We don't have these available, so that's what we have to work with. Nevertheless, knowledge about the soft tissues could be very interesting, especially if we had knowledge about the brain. If we knew what the brain would look like, we could make inferences about behavior, about intelligence, about a variety of different properties. So how can we actually find out more about the soft tissues, in particular about the brain of Allosaurus here, if we only have the bones available? So we can take a literal look inside the head of Allosaurus. That reveals that we have a large cavity where the brain was located once or when this Allosaurus was alive. This brain cavity, or endocranial cavity as we call it, that's the space where the brain was located. It's surrounded by the brain case bones. This region of the skull is a very, very sturdy and compact region. It protects the brain, it houses the brain, and it also communicates via several holes with other organs. You can see these holes here. So that's where the cranial nerves from the brain go through the brain case to innervate different organs. So based on this general morphology of the brain cavity, we can already make some assumptions, some inferences, how the general brain shape might have looked like. And that's not something new. That's what researchers 100, 150 years ago have done. So I'll briefly talk you through the historical background of paleoneurology, or the study of the brain and associated nervous and inner ear tissues in fossils. So very similar to what I've just shown with the Allosaurus skull, researchers 100 years ago asked themselves the same question. So if I have a brain case, in this case of Tyrannosaurus, and cut that open, make a vertical cut and look at the brain cavity, is this something which I can use to infer the brain morphology? And the traditional approaches were mostly artistic, interpretive. So in this case, an artistic 2D drawing of the brain or a cast of the brain cavity has been produced to show what the brain might have looked like. And this was a common approach at the beginning of the last century. So a variety of different fossil animals, from fossil birds to different dinosaurs, have been studied that way. Their brains inferred and reconstructed in a similar manner. One of the researchers which was pioneering in this approach, I want to name Tilly Edinger, who not only studied the brain morphology of specific species, but actually looked at the evolution of the brain. So for example, what happens with the brain during the evolution of horses from very small beagle-sized animals to normal horses we know today. So what changes not just in the body morphology, but also in the brain morphology. In some rare cases, we actually have, as paleontologists, fossilized brain available, or similar to fossilized brain. This is the brain cast of Allosaurus. Just to orientate you here, that's the front part of the brain, that's the hind part of the brain, so we're looking at it from a right aspect. This is not fossilized brain itself, but it's a sedimentary cast of the brain cavity. So this happens when sediment fills a skull, the sediment solidifies, then the softer bone around this solidified sediment is dissolved away, and then what remains is a sedimentary cast of the brain cavity, the inner ear, and associated structures. As you can imagine, these examples of such spectacular preservation are very rare. We have a few examples, for example, this Brachiosaurus brain here. So Brachiosaurus, one of these large, long-necked sauropod dinosaurs. Again, we have the brain here. The top end is here, the rear end is here, and the inner ear of Brachiosaurus here on the right-hand side. As I said, these are fairly rare occurrences of these exceptional preservations where we can get actually an idea of the brain cast. But for researchers at the beginning of the last century, this wasn't obviously enough, and also the interpretive approach of reconstructing the brain wasn't good enough. It was attempted to do this in a more quantitative way. So one of the researchers, William Solis, who was later a professor in Cambridge, he invented this machine, a so-called grinding machine. And that works in the following way. So you would take a fossil skull, put it in that contraption, turn the crank a few times, and this disc here at the bottom would ground away parts of the specimen, a few millimeters. Then you would take out the specimen, the freshly exposed surface you could replicate either by drawing or as small layers of wax. Then you repeat the process, the skull goes back into this contraption, the crank is turned a few more times, more of the specimen is ground away, and a newly freshly exposed surface is then exposed. And by this way, you can take a physical cross-section through the whole skull. So this is the first example of physical tomography, where you divide a whole specimen into a number of different slices. And these slices can be reproduced, for example, in wax, as has been done here by Solis with this Paleozoic fish skull. So you can see these very fine layers. These are replicates of the ground away surface and then glued back together in wax. and the corresponding brain of this Paleozoic fish as well. Here we have a 3D example, and you can see that this method really produces a high-resolution brain reconstruction. So it's very good to capture all the detail, but I'm sure some of you have already noticed the big disadvantage with this. So if you put a skull in there and then grind it away bit by bit, obviously what you end up with is a handful of dust. So if I went to the Natural History Museum today and asked them to give me one of the Tyrannosaurus specimens because I want to sample it in this way, they would probably quickly show me to the door, because obviously this is destructive sampling, and if we have rare specimens, we don't want to destroy them. So researchers had to come up with different methodologies to reconstruct the brain morphology, to get a cast of the brain cavity. In this case, this brain reconstruction of this dinosaur was done in latex. So latex would be painted inside the endocranial or the brain cavity, and once this solidifies, then it can be taken out as it's flexible, and you would get a cast of the brain cavity. But in comparison with the two previous slides, you can see the resolution isn't that good. Not all of the internal details are captured, so this, although non-destructive, is not the best approach to get the full detail of the brain reconstruction. And finally, when we get to the 21st century, we get more computer technology available. So this is the first example of using computer tomography to reconstruct a dinosaur brain. Computer tomography or CT scanning you might be familiar with if you have been unlucky enough to break a bone, and in the hospital you were exposed to these X-rays from different aspects, which would allow then to reconstruct a three-dimensional image of the scanned specimen. The same was done here, and I appreciate that doesn't look like very much, but at the time that was a bit of a sensation. So this is the first example of how a brain could be reconstructed using modern computer technology. That was about 20 years ago, and since then a lot has progressed in terms of digital visualization, virtual reconstruction, and I'll briefly talk you through how we can recreate the brain morphology using computer tomography. I'm showing the first example here using a modern bird. This is a common buzzard, which we CT scanned. And here on the left-hand side, you see a first slice through the specimen. So these are numerous cross-sections through the specimen from the back of the skull towards the front. Here in the middle, that dark gray area, that's the brain cavity, surrounded by a very thin layer of lighter gray bone. That's the brain case, and a lot of muscles around the specimen. Correspondingly, so we have here the cranial cavity, the brain cavity, and if I move through the CT data set through all these slices, I can highlight the regions of interest, which I want to particularly show. So for example, in blue, the brain cavity or the brain cast, then in this reddish color, the inner ear, and in yellow, the different cranial nerves. And if I do that successively for each of these slices within the data set, I can slowly reassemble, recreate this whole 3D morphology of the brain. So create a digital cast, a digital endocast, as we call that, of the brain morphology. And that then allows me to use this 3D model in virtual space to study the brain and make inferences. That obviously works also on fossils, so we could do the same on our Allosaurus here, rather than just doing interpretation of what the brain might have looked like on the basis of the endocranial cavity. We can actually use CT scanning and this virtual reconstruction technique to recreate the brain of Allosaurus. Some of you might have noticed this doesn't look really like a brain, or not what we would expect a brain to look like, especially if we think about a human brain. So I'll talk you a bit through the fossil brain anatomy, and brains can look very variable depending what kind of group we're looking at, what kind of animals we're looking at. Just to orientate you in terms of the colors, by convention, the proper brain itself is mostly shown in blue, and this reddish pinkish color, that's the inner ear, and all the neurovasculature, so blood vessels, nerve canals, things like that, are shown in yellow or in reddish color. In comparison to the human brain, this looks very, very different. So just to orientate you, I'll have this reconstruction of a human brain here in the corner, so you can see the corresponding region of what the Allosaurus brain looks like and what the corresponding region in the human brain would be. So if we start our journey through the dinosaur brain, starting at the front, we see this region here, the so-called olfactory region or the olfactory tracts. This is the region in the brain which is responsible for the sense of smell, which houses the different sensory cells for smelling. And there's a rough relationship between the size and the capability to discriminate between different smells. Just in comparison, that region in the human brain is very, very tiny. Not surprising, because yes, we know that the human sense of smell is not the best, especially compared to other mammals, but also compared to dinosaurs. And if we look at different dinosaurs and also at the natural endocast, here a natural endocast of Tyrannosaurus, we can see that in some of these dinosaurs, this olfactory region is really, really massive. And although there might be a bit more sediment contained here than is actually the brain, if we compare that with a computer model created with CT scanning, we see that this region is really, really extensive in most of these predatory dinosaurs, and that in particular T-Rex might have had a very good sense of smell. So if a famous Hollywood paleontologist tells you, don't move, T-Rex can't see you, that might be correct, but I'm pretty sure T-Rex could smell you, so it's a good idea to run away nevertheless. If we move a bit further through the brain, we see this prominent region. It's the so-called forebrain region, or the cerebral hemispheres. We can see the two spherical shapes here to the left and right of the brain. In the human brain, this is what we usually associate with a brain morphology, so these walnut-shaped, very complex structures which overlap the rest of the brain. In dinosaurs, this region is fairly small in comparison to the rest of the brain, but it's very distinct, so we can clearly discriminate it from the rest of the brain. And that's the region where the cognitive capabilities are located. So we'll try to stay away a bit from the term intelligence, because intelligence is obviously a lot more a very complex subject and difficult to test in living animals as it is, even more difficult in fossil animals. So we'll come up with the term cognitive capabilities. And again, there's a rough relationship of how large this region is and how good the cognitive capabilities are. And paleontologists have developed a kind of a ratio or a quotient how to measure this forebrain region and this cognitive capability. So based on observations of modern animals, there's a certain ratio between how large the brain mass is in comparison to the body mass, and we can put that into context with the fossils. So how large is the fossil brain in comparison to what we would expect the brain to be like based on the body mass? If we look at this ratio, this quotient, and dinosaurs and their modern relatives, crocodilians on the one side, birds on the other side, you see that crocodilians are fairly low on that scale, so they are probably not the smartest, not the poets and engineers and inventors, if we want. Whereas on the other side, so the birds, they have a fairly high ratio. Not surprising, we know that several bird groups are very, very capable of cognitive achievements. So parrots, ravens, crows are capable of problem-solving strategies and similar. So yes, the cognitive capabilities in birds are very high. Our dinosaurs fall somewhere in between, so Allosaurus in particular, somewhere in between. The large-bodied sauropod dinosaurs, they have a very low quotient. That might be due to the large body size and the small heads. Whereas some of the dinosaurs closer to modern birds or leading to the bird lineage, they have a very high quotient. Some of them might even be higher than modern birds. So we can assume that some of these predatory raptorial dinosaurs might have actually been quite clever. In that context, usually someone mentions Stegosaurus. Stegosaurus is notorious about having a very, very small brain. So Stegosaurus, this ornithischian dinosaur, very characteristics, about 5 meters in size, 5 tons heavy. If we look at the brain of Stegosaurus, it's very, very tiny. And usually the comparison is made that Stegosaurus has the brain the size of a walnut. That's absolutely not true. It's 2 walnuts. Maybe we can squeeze a third one in there. But yeah, it's not the largest. So based on the body mass, the head size, it's a fairly small brain. In this context, I usually show my students this image, and they're a bit flabbergasted because they don't know what it is. So if you started playing around with computers in the 90s, like I did, this was the state-of-the-art storage medium at that time. It could hold 1.4 megabytes of memory. Compared to modern storage media, obviously, this is ridiculous, where we have several orders of magnitudes more storage capability. But if you compare the size, obviously, we have a just physical size, a larger object and a smaller object. So the size itself is not necessarily representative, but how you use the size and how densely you pack information on it. And the same is true, apparently, for brains, at least from modern studies, what we can tell about bird brains and mammal brains. Although birds have smaller brains, leading often to the misnomer bird brains, that's absolutely not true. We see, although the volume is smaller, they pack more brain cells, sensory cells within the same space compared to mammals, where we have a larger brain, but fewer sensory and brain cells. So the resolution of how this volume is used plays a role as well. If we move a bit further through the brain, we get to this region. This is the mid and hindbrain region. And fossils, it's a bit difficult to discriminate because it's lost somewhere here in this endocast. It's not a clearly demarcated morphology. That's where the sense of locomotion, the motor control, these kind of things are located, but difficult to get a grasp in fossil brains. And the human, it's a decent size, but basically it's overlapped by the forebrain region, so we can't see the maturity of the mid and hindbrain. Much more interesting to get an idea about locomotion, the sense of balance, the sense of hearing is the inner ear. So this pinkish structure here compared to the human condition, very similar, slightly different orientation. So that's where the inner ear sits. The inner ear, obviously, responsible for the sense of hearing, but not only for hearing, but also for the sense of balance and equilibrium. And there are 2 parts to the inner ear. So we have the top parts. There we see these semicircular canals. These are 3 canals which are aligned with 3 spatial planes. And those canals are hollow, and the fluid is moving around these canals. And when we move our head, or any animal moves its head, then the fluid inside these canals moves around, dislocates little sensory hairs, and this dislocation of the sensory hairs then makes an impulse to the brain, and that's how movement is registered. That's why we have these canals orientated along the 3 spatial axes. That's how we can perceive 3-dimensional movement and information. You mostly notice this when you are on a roller coaster or similar, because then you have lots of movement. The sensory hairs in these canals get dislocated very quickly in different directions in quick succession. That leads to a sensory overload to the brain, and then you feel the effects by getting dizzy or queasy. That's what happens in these canals. The lower part of the inner ear, that's where the sense of hearing resides. And again, there's a rough relationship between the length of the so-called cochlear duct and how many sensory cells can be packed into that. So the larger this region, the better the sense of hearing. If we calculate that for different dinosaurs, including Allosaurus here, so we can calculate the best frequency of hearing. That's the frequency where sound can be perceived at best. So that's around 500 to 1,000 hertz. and the maximum hearing range or hearing limit. So that's how many different frequencies can be discriminated, going from 2, maybe up to 4 kilohertz in these dinosaurs, compared to humans where we can maybe reach 18, 19 kilohertz. This is not that impressive. So we have a lot lower capabilities of discriminating different sounds, about a quarter or a fifth of the human condition. But we can also do a bit more in terms of inferences about behavior and biology with the inner ear. So if we look at this semicircular canal, which is orientated parallel to the horizontal plane, it's the horizontal semicircular canal. If we orientate that with the ground, that gives us an idea about the typical or habitual head posture. Because in that condition, there's minimal dislocation of the sensory hair. So that's how, for example, Allosaurus would hold its head in a typical position. Compared to humans, yes, that makes sense. That's how we usually hold our heads straight forward in a typical alert or habitual position. So we can use the orientation of this semicircular canal to infer the head posture of different fossil animals. You would think, well, most animals have the head level and straight on. Yes, that's probably a good rough estimate. Let's see what we find for different fossils. So in this case, we have a sauropod dinosaur, or again, one of those long-necked dinosaurs, Nigersaurus. And usually we would assume that sauropods have the head at the end of these long necks, somewhere high in the treetops, in the crowns of the trees, feeding on different vegetation. If we reconstruct the brain of Nigersaurus here, so that's where we have the brain and the inner ear. If we align that with the skull and with the ground, we see that's a very downturned head posture. So the inference here is that this dinosaur is actually not holding its head high up, but rather lower to the ground. So in this case, this dinosaur has been inferred to use the long neck to increase the range when browsing for vegetation on the ground, rather than using that in the crowns of the trees. That also works for marine animals. So in this case, we have a so-called placodont. This is a Jurassic reptile, mostly marine. And if we reconstruct the brain here, put it inside the skull and orientate the canal with the ground, we see, again, a downturned head posture. And consequently, this animal has been reconstructed as mostly looking downward when diving towards the seafloor and feeding on hard-shelled organisms, mollusks, and brachiopods. And that also makes sense if we look at the dentition of these placodonts. These are very, very sturdy, broad teeth, which have been inferred to be used to crush shells and mollusks, brachiopods, things like that. So that aligns nicely with the inferred posture of the whole animal, or of the head in this case. A third example, so not just a level or downturned head position. In this case, we have one of these weird Jurassic reptiles, Proterosuchus. If you wonder about the tip of the snout here, that's natural, so that's not taphonomy, not deformed or dislocated. We have several specimens which show exactly that morphology. If we reconstruct the brain, and this is work done by my former master student, Emily Brown. If we look at the brain and position the head accordingly, and we see this is not a downturned, but in this case, an upturned head posture. So what was Proterosuchus doing with this head held upwards or looking upwards? Well, it's a marine predator similar to modern crocodilians. Most of the body was probably hidden below the surface. Just the head would be above the surface. The nostrils would have to be above the surface so it could breathe. And that makes sense that in this position, yes, it could just look out from the water, but the rest of the body would mostly be hidden. So we could and can reconstruct a lot of biological and behavioral properties using these brain reconstructions, and my research group and I have reconstructed the brains for a variety of fossil animals. Just here a few examples, from dinosaurs to birds, turtles. Work from two of my PhD students, Luke Mead here, who reconstructed the brain of a cynodont, and Alejandro Serrano, who reconstructed the brain of different crocodilians. So we can use these brain reconstructions, these virtual reconstructions, to infer a lot about individual species, individual animals, but obviously we could do a lot more, and that's where things are becoming interesting if we're looking at how the brain changes either during individual development or in the course of evolution. So let's look at what happens in terms of development, how the brain changes during the growth of an individual animal. In this case, we looked at another dinosaur, an ornithischian dinosaur named Dilophosaurus from the Jurassic of Tanzania. The good thing with this species is that there are lots of different individuals of different age classes. So we have a juvenile, nicely preserved skull. The animal is was about 2 years of age, and we have a fully grown individual of about 13, 14 years of age. So we reconstructed the brain anatomy of the juvenile here and attempted to do the same thing with the adult. That was a bit more complicated because we didn't have a nicely articulated skull, but all the skull bones were as different pieces, little jigsaw pieces, if you will. But not a problem if you're using digital technology, so you can simply reassemble the whole skull, or at least the brain case, which is relevant for these kind of reconstructions, and then perform a similar brain reconstruction as before. So that we now end up with two models, one for the juvenile specimen and one for the fully grown specimen. And you can see, although these are the same species, the brains look very different, and a lot happens during the growth of an animal. In this case here, in about 10 years, maybe a bit shorter. We don't have that resolution. And if I animate that, we can see how the brain changes from the 2-year-old individual to the fully grown individual. So we see that the mid-hindbrain region here at the back, that increases massively in size. The forebrain increases in size. The cerebral hemispheres get larger. The olfactory tracts get larger. One interesting thing which doesn't change if you look closely, that's the inner ear. So the inner ear doesn't change very much. And if we calculate the properties and inferred capabilities for this species, we see that a lot of the parts of the brain change very considerably. So some of the brain portions increase in size by 50 or more %. What doesn't increase, though, is here the hearing capability. So just by a few percentage, this is not significant. That led us to infer that the juvenile could hear the same sounds as the adult specimen. And based on that, we inferred further that communication along this frequency range was very important. So if they could hear the same things, they probably could produce the same sounds, and that might be an indication for a closer social structure, so that the parents would be able to communicate with the juveniles, that they would live in the same group. And actually, evidence from the fossil bone beds where these different individuals have been found in close proximity would further indicate that, yes, these dinosaurs lived in a very close social structure, and that vocal communication was very important between the juvenile and the adult specimens. So we could look at what happens during individual development of an animal, but at the same time, we can also look at what happens during the evolution of a large group, what happens to the brain in terms of ecology. Is brain morphology linked more towards how similar different species are, how closely they are related, or is brain morphology controlled by external factors? So where the animals live, how they function, what kind of requirements that they have. So we looked at that in several studies. First of all, we looked at the phenomenon known as convergent evolution. Convergent evolution indicates that two different species which are not or only distantly related emerge with a very similar morphology. So for example, we have this group known as phytosaurs, again, one of these Jurassic groups restricted to the Jurassic, superficially similar to crocodiles. They're also distantly related to crocodiles, but are at the base of the crocodile family tree, but superficially very similar to a modern gharial. So they both share the long snout, the skull morphology, and we were interested, so if they share the same skull morphology, does that mean they also share the same brain morphology? And if that's the case, then that would indicate that superficial similarity also is imposed on the brain. So we reconstructed the brain morphology for two of these phytosaurs and a modern gharial, as done before, and compared those brain shapes, and we see that the phytosaurs between each other, they are very similar, so they have this very elongate but narrow brain shape, a very small forebrain region, not very distinct. If we compare that with modern crocodiles, we see that this is very, very different. Here we have a more compact brain shape with a very prominent forebrain region. The cerebral hemispheres are very, very extensively developed. So clearly a difference between the crocodile and these phytosaurs. And we looked at more members of these two groups, and we see that the morphology for the gharial is actually very similar to the rest of different crocodilians. So they all share this morphology. On the other hand, if we look at the phytosaurs, they all share this very elongate but narrow brain morphology. So in this case, we can clearly say that how closely different species are related controls how the brain looks like. So if you are a crocodile, your brain will look like this. If you're a phytosaur, your brain will look like this. That might not necessarily be true for all animal groups. So we tested that a bit more, or we are in the process of testing that a bit more, for example, looking at saber-tooth cats. So here we have three different saber-tooths. Again, they share a superficial skull morphology, and the question is, does the brain follow that as well, or will the brain be very different? And in this case, this is an ongoing study done by one of my third-year students, Thomas Henton, who has done an excellent job in reconstructing these brains. And we can see preliminary in these results that the brain actually looks very different. So in this case, the closeness in relationship does not seem to indicate that the brain morphology would look the same, but other factors might come in here. Ecology, how they live in social structures or not, might influence the brain shape, and we can see that here, based at least on these preliminary results, that there are differences. In this case, not controlled by how closely they are related, but how they interact with the environment, how they interact with the ecology. So based on that, we also looked at can we use these ecological signals in the brain to infer the living conditions of fossils. And in particular, we were interested in the origin of turtles. Turtles, or their origins, are still shrouded a bit in mystery. There's a debate whether the first turtles originated in a marine environment or on a terrestrial environment. One of the first turtles with a proper shell, we have here Progonochelis from the Paleojurassic of Germany. This turtle is already quite developed, so it is a bit further than the absolute turtle origin. But still, we were interested, could the brain morphology tell us something about where turtles come from initially? So we looked at modern turtles and wondered whether brain morphology is actually different when we compare where these different turtles live. And we see that marine turtles are very different from freshwater turtles, from terrestrial turtles, and also from boring turtles. So we reconstructed the brain morphology of this first turtle, Progonochelis, and looked at the brain and compared that with the rest of the modern turtles. And we see, yeah, just by eyeballing, that looks very different to what we have in modern turtles. But we wanted to quantify that a bit more, so we used a shape analysis approach. So this is similar to a face recognition approach. which compares shape differences more quantitatively. And we looked at different species which were aquatic, so either marine or freshwater, which were terrestrial, so living on land, and which were living below the ground, so fossorial, digging or burrowing. And we compared the morphology of the brain of Progarnachilus with the rest of these, and in the different analysis we found that Progarnachilus actually falls outside of that of modern species, turtles and other reptiles, but clearly somewhere on the terrestrial side. And in the second part of the analysis here, we find it falls somewhere within the terrestrial fossorial range, so a terrestrial or burrowing turtle. So based on that, we inferred that the first turtles might actually have lived on land, and not only that, they might have been actually burrowers. I should mention that turtle origins are highly controversial, and this is probably not the last word spoken about this topic, but it's nice to see that we can use inferences on soft tissue morphology to elucidate these evolutionary patterns a bit more. So far, I've shown you quite a lot of what we can do with these virtual reconstructions, but obviously we can always do more, or there are always outstanding challenges, and I'll briefly talk to you for the rest of the talk of what the future might hold in terms of paleoneurology. So one of the things I very rudely gloss over is that obviously if we reconstruct the brain morphology based on the brain cavity, this is the cast of the brain cavity, that doesn't necessarily mean that the brain always filled the brain cavity fully. There are other soft tissues within the brain cavity, brain fluids, the meninges, different things. So the brain is suspended in these, and based on which group we look at, the brain might have or might not have filled the brain cavity completely. To get a grasp of that is very difficult, but there's a group in the US working on that, trying to elucidate what part of the brain, the proper brain, is actually contained in these endocasts. Is it much smaller? Is it close to a 1-to-1 ratio between endocast and the actual brain shape? So this is something which could reveal a lot of new information in the future and would allow us to get a better grasp on different brain regions. The other thing which I've shown in the course of this talk, whenever we reconstruct the brain morphology, actually we are dependent on well-preserved fossils, complete skulls, complete brain cases. These are rarer than you would think, so we are dependent on good preservation. And whenever we reconstruct brains, we usually do that on a single specimen. The question here is that specimen and its brain morphology actually representative of the brain shape of the overall group? So this is another challenge we have to tackle in the future. So in this case, the first case study here was done with one of my students, Charlotte Bird. She looked at the brain morphology of different specimens of the same species of a proto-mammal, a cynodont known as Rhinaxodon. And we can already see that the brain morphology varies a bit between individuals of the same species. So probably in the future, we need to be careful of how much we interpret into a single brain reconstruction if we don't know the variability in shape and morphology of these brains. And with that little outlook, I thank you all for your attention. Lots of colleagues to thank who contributed to different studies. My research group and my busy and very diligent students who contributed a lot to this. Various funding bodies, obviously you for your attention, and I'm happy to take questions if we have time. Thank you very much.
Thanks. We definitely have time for some questions. So if you could just wait for the microphone to get to you before speaking. Do we have any questions?
Hi. Is any work being done on pterosaurs, flying reptiles?
Yes, there are a few studies on pterosaurs. They are quite interesting, and so far that the brain morphology is in many aspects very similar to that of birds. because they lived in the same environment. So again, they have to deal with complex three-dimensional movement, and in particular, the inner ear shows an adaptation to that. So that's a good example of convergent evolution between birds and pterosaurs in the brain as well.
Thank you.
I don't know if this is working or not. In the human brain, there's two distinct lobes. Do you find the same in paleo brains?
It's difficult. So in hominids, you can possibly see that. The further you go back to dinosaurs, birds, reptiles, it's difficult to see the finer details within the brain. So we can't distinguish necessarily between different regions. We can get the general morphology for mid-hindbrain, but a higher resolution than that is getting difficult because we don't have that resolution from the endocast. Not yet, yes, that's true.
Thank you. Is there any similarity in the brains between early mammals and the mammal-like reptiles?
Yes, so we see that mammal-like reptiles, Dimetrodon, something like that, very similar to cynodonts, which are on the lineage towards modern mammals. As soon as we get to proper modern mammals, we see changes in the brain morphology, that forebrain region gets a lot larger and more complex. Also, the inner ear changes, especially that region which is responsible for the sense of hearing. So what I've shown in dinosaurs, we have a very simple straight canal for the hearing cells. In mammals, that's very complex and spiral shaped, which increases the size and the volume, but in a smaller space. So that allows mammals, for example, to discriminate between a wider range of frequencies. So that's how humans can hear up to 18, 19 kilohertz, whereas dinosaurs couldn't.
Do you have any other questions? Please, yes. Oh, sorry, Amelia's the one.
The Stegosaurus has a very small brain of 2 walnuts. Does this mean that it has a high density of sensory cells, or is it just stupid?
The truth lies probably somewhere in between. The newest study I've shown with the density of the brain cells between birds and mammals, that's probably something very newly acquired in birds and mammals. Probably it doesn't go back along the bird lineage towards dinosaurs. So yeah, Stegosaurus might have not been the most clever one.
Why are the semicircular canals so intimately associated with the cochlear canal?
Good question. I must confess I have no idea how that emerged or evolved, that two more or less distinct sensory systems are combined in the same organ. So it might be that in fish we have little crystal-like structures which are both responsible for hearing and equilibrium. It evolved from there and split up into 2 distinct regions.
Good one.
Hello, is it working? So with Allosaurus, from what we can see with the brain, what does it suggest about its behaviour?
So the question was, with Allosaurus, what can we suggest about behavior of Allosaurus based on the brain? We can clearly see that it has a large olfactory tract, so the sense of smell must have been quite good. Actually, in theropod dinosaurs, the sense of smell seems to be quite good in general, so they could discriminate between a wide variety of smells. That probably also is true, or as far as we can see in birds. Birds actually have a better sense of smell than we would assume or we would typically associate with. So with theropods and Allosaurus, it probably was a fairly keen hunter with a good sense of smell and would probably rely quite a lot on sense of smell rather than, for example, hearing, because the hearing capabilities is very narrow towards the lower frequencies. So rather than the hearing, it might have been the sense of smell, possibly vision, but we can't tell that much from the brain about the vision.
So mostly a solitary animal then?
Difficult to discern from the brain because that region for pack hunting or solitary hunting is not that developed in the dinosaurian brain. We could possibly see it in the saber-tooths, and that study might show us something about whether different saber-tooth species might have been pack hunters. But in the dinosaurian brain, the forebrain region is not that developed that we could actually tell something about this.
Thanks. In the back.
Is it possible to use the same techniques that you've used in the brain to infer about other organs, such as the skin?
It's possible, for example, for muscles. So whenever an organ is enclosed by bone, so we need some kind of constraint, the bony structure, the skeleton, which gives an idea how large the empty cavity in between or the sensory organs, the soft tissues were in between. So it works with muscles. For example, the jaw muscles, we can reconstruct the jaw muscles and then find out more about how powerful a bite of a dinosaur was. The skin is a bit difficult because it's external to the skeleton, so we don't have necessarily an idea how much the skin was constrained by the skeleton. There are a few examples of skin preserved as molds or as casts and fossilized skins. We could possibly tell a lot more about the skin structure from preservation rather than reconstruction in this case.
Hi, Stephan. So the relative size of these brains to human brains wasn't really discussed. So I'm kind of curious as to how big are these dinosaur brains and how does that kind of relate to the size of the dinosaur as well?
Yes, that's difficult. So these experimental observations of how brain size scales with body mass in modern animals, that gives us an idea whether this is a linear relationship or not. So we can use that also for the fossils, and then would we expect that the fossil has a higher or lower brain size than modern animals? Compared to humans, the absolute brain size is probably a bit larger than in many dinosaurs. Might be similar to T-Rex, where we have a skull of about, 80 centimeters, a meter or something like that. And just by having a larger skull, you have a larger brain, and then you have, in absolute measures, a larger brain. Relative to body size, obviously, the human brain is quite large. So, and especially in mammals and in humans, we have the highest brain-to-body mass ratio in most of the species. So dinosaurs, birds, crocodilians, things like that, the further we go down the evolutionary tree, we get a smaller relative brain size.
Using these brains, how much can you tell about their environment, and then how can you compare it to our environment to see the changes that it has gone through?
Yes, very good question. So the problem with the brain is during the growth of an individual, the brain changes quite a lot. But once it has fully grown, it's not as flexible, so it wouldn't show a signal from when there's changes in the environment, seasonal changes or catastrophic changes possibly. With regards to climate change, we can't really see that in the brain because once it's fully developed, nothing changes substantially. But it would be interesting if we could reconstruct that. But the brain morphology is not a good proxy for that.
Thank you. Can you tell how thick the optic nerve is, and is that any use?
In some of the specimens, yes, because the optic nerves need to exit the brain case, and based on the size of these foramina or these holes, we can tell how large the optic nerve is. And it's usually the largest of the 12 cranial nerves. And in some of the fossil specimens, it's really dominant. In many of the theropod dinosaurs, it's very dominant. We can get an idea how large it is, yes.
I think if that's come to the end of these great questions, please join me in thanking Stephan one more time.
Thank you. That's right. Good afternoon, everybody. Excellent. Liquid turbines. Okay. In the undergrad course, dedicated paleo at the moment 12 in the first year. So we've grown quite a lot, yes. Yeah, when I started in Birmingham, there were 2. The following year we had 8, and the current first years are 4. Okay. Right. Yeah.